Okay, so yeah, as you heard, I don't know if I have a cursor. Oh, yeah, I do. There's two of us have a cursor. That's interesting. Okay. Yeah, so I'm going to tell you today a lot about the work on OME. I won't tell you too much on our commercialization of that through Glencoe software. If anybody's interested, happy to talk about that. I will say it's interesting. You're working in an academic environment where trying to get data sets to drive the directions we're going is great. In a commercial environment, boy, oh boy, can you get your hands on data. And that's actually that, hey, you have use cases, you have a sort of practice there. We were talking over lunch about Welcome Leap. That's a completely different talk, but for those of you who know something about the United States DARPA, the Defense Advanced Research Projects Agency, credited with inventing the internet, GPS, things like that. So the idea is, can you take the methods for driving innovation that have been successful at DARPA and bring them to the life sciences? So I'll have to talk about that if you're interested afterwards. Okay, so I constructed this talk, so I will confess I'm starting to get old. And so I've constructed this talk a little bit about how I got here and just the story of everything that we've done. So hopefully that's helpful, especially to the younger members of the audience. So first of all, a standard commercial disclosure. It's noted I'm the founder and CEO of Glencoe Software. So Glencoe takes OME tools and has and provides commercial licenses for them. So we work a lot with academic medical centers around the world, biotech and pharma. So obviously there's a financial conflict there, but you should just be aware of that. Okay, so, okay, the story starts, graduated from university, I was a bike racer, and I spent several years racing around Boston and California, going fast down hills and all that stuff. Eventually dawned on me that that was probably not a long-term strategy. So going headfirst into things at high speeds has a certain limitation. So I was working as a technician in a laboratory in San Francisco at UC San Francisco. And this was a time that something called site-directed mutagenesis had appeared. So I don't know how much this will mean to you guys, but at the time, the idea that you could take a piece of DNA, a cloned piece of DNA, make a mutation, change a base pair, and that would convert, that would then be expressed to a protein that had a different amino acid sequence, and then you could then assay that, was extraordinary. And the first examples of being able to do that and to change enzymatic structure and enzymatic activity had just happened. And this was extraordinary. It felt like we could now finally understand how proteins actually work. So that's what I was going to do. And I was interviewing in a lab with a guy by the name of Dave Agard, who had just published this incredible paper on re-engineering bacterial protease. And we were talking about various experiments, and he said, I have this other side of my lab. I don't want to hear about it. We do a lot of cell structure, cell and tissue structure. I just thought that's odd. No idea what that's all about. And it was actually fairly late in the evening. It was about 8 or 8:30. We worked fairly late. And I was hungry, but you don't say no. And so I said, okay, sure. And he started talking about, so he started talking about the fact that they had, so his lab, along with a collaborator, whose name was John Sadat, they were the first people in the world to have a CCD camera and hook it up to a microscope. It's the first place in the world that was taking digital images of cells and tissues. Now, you guys take that all for granted. Now, I'll just say that gives you a sense of how old I am, but also, to land in a place that was starting to take this data and just for the first time be able to work with these images and start to do experiments was incredibly exciting. And so I was taking, in my PhD, we were taking 3D images of cells and some tissues, but admittedly cells. And so I got my PhD on the back of taking movies like this one. So this is it doesn't project too well, but this is a Drosophila embryo. On the scale of this image, you have to imagine that the embryo goes far out into the lobby and far out the windows. But this is the syncytium of the Drosophila embryo. And you see these nuclear divisions. Now, you guys are used to seeing fluorescent images of time-lapse images, but we didn't have GFP. This is 1989, 1990. We didn't spell GFP. So you had to purify the proteins, label them with a fluorophore, inject them back. So it was actually very technical, et cetera, but it was incredible, and did this and a few other proteins. And I collected in my PhD about 10 live imaging sequences. Okay, and on the back of that, I got a PhD. So compare that to what the number of images that John worked with to get his PhD. So, this was very, very early days, but imaging was certainly pretty exciting. And I went through a postdoc at UCSF and at Harvard Med School, then for various reasons moved to Dundee. And we started doing time-lapse imaging of cells and tissues. Automation was getting better. Cameras were getting cheaper. GFP fusions were available, et cetera. And we're starting to publish papers with now hundreds of time-lapse sequences. And so what we're starting to run into this data problem. So that the number of images we had, how we were going to process them at scale, how we were going to connect various analytics, et cetera, were becoming limiting. So this idea of data management was becoming a problem. And so we naively decided to work on that problem. And what we realized is that that problem is ubiquitous. Everybody was having this problem. Everybody was struggling with how they were going to handle their data, how they were going to contend with making the analytics work to get the measurements, and also this idea of collaboration. So I would have some time-lapse data sets, and I wanted to collaborate with somebody else in a different institution. How would that happen? We had no way of doing that other than copying on disks and transferring them. But even then, formats, et cetera, would block it. So that was the genesis of what we started working on. And looking around, you could see that imaging data was everywhere, and more people were using imaging for more and more different types of measurements, and more modalities were coming online as people were getting more, incredibly ingenious and inventive about all of the different ways you could use imaging. And so, the problem just kept growing and growing and growing. And so, in a vision, you could start to imagine that, well, gosh, you want to be able to access all of these data, and ultimately, you'd like to, for example, publish them online so that people could share and work with these data sets at scale. And maybe one day you'd like to build an online database with all of the data annotated and everything available for everyone in the world to see. That was the vision way back in 2000, and that's what we've been working on for better than 20 years. So the project I'm going to tell you about, it's called OME. And this is an open source image informatics project. So passionately open source, everything that we build is publicly available. And it's available more or less as soon as we generate the work. So we don't wait, for example, for publications. We take the responsibility of delivering these tools to the community, but also take the responsibility of trying to build tools that can support our work and others' work, but also make that as innovative as possible. So to give you a sense of what we do, we don't do anything that wins Nobel Prizes, that's for sure. So imaging has really taken off over the last 10 or 15 years with lots of really exciting new data acquisition systems and data modalities. We don't do any of that really exciting work. But we do get quite interested in the structure of the data, the binary data, the raw data, and the metadata, and we try to find out ways that we can write that down to make it as accessible as possible. And what we've been really focused on over the last 20 years is connecting all of these different parts of the sort of the peripheral part of this diagram. And so we sort of define success when, you know, an incredible amount of analytic tools that are already developed can access data coming off of new imaging systems. So without anyone ever meaning to do so. So this idea of interoperability or connectivity between analytics, data, et cetera. So, you know, here you will understand that here we're talking about APIs, et cetera, but you can think of these as as ways of connecting data when I give talks to biologists, the central broker or something like that. And by no means do we think that we can be the only entity here, but really what we're trying to do is lower the boundaries between sort of the left side of the diagram and all of the other parts of parts of the slide. And we want to do this for individual labs, for institutes, and ideally for national or global repositories. And the aspiration was to be able to build tools that could work at any of those scales. And so that's what I'll try to show you today. So these are names of software that software various tools that we release. They're all open source. And you'll see a few of these names as we go through the talk, but I'll focus today on bioformats, OMERO, IDR, and I'll finish up with a little bit of a discussion about OME-TIFF. I should say, please stop me. I'm happy to take questions as we go on. Okay. Yeah.
The markers with the environment, what does it stand for?
Yes, thank you. I should have said that. So just OME stands for Open Microscopy Environment. And we've had lots of people say to us, well, what the hell did you mean by that? What a terrible name. So open, that was easy. Microscopy, that was probably a mistake because we started out in the world of biological microscopy, but we've definitely moved far out of just sort of traditional microscopy. Some people have said, well, you should change the name over to metadata. Yeah, but metadata is important, but it's part of the story. And a lot of people have objected to environment because, well, what does environment mean? It was, you know, a place or a locale where you could work. But yeah. So we just refer to it as OME. Okay, but it's a good question. Thank you. And I should have pointed this out. Sorry, the website is there. And the final thing I should say before I go on, so probably don't need to do this in this audience, but I definitely get asked this question. And I get asked, well, is OME free? And as the guy who has to pay the bills and write all the grants, OME is definitely not free. It costs in grant terms something on the order of 20 million pounds. So it's definitely not free, but obviously the technology is freely available under either BSD or GPL licenses. And if you want, we can talk about the choices that we make in those licenses. Okay. So another way to think about what we do is building interfaces, as we said. And so kind of starting on the left side of the slide, most modern research institutions, either academic or industrial, will have many different imaging facilities, and these will be technologies that are dedicated towards the research interests. Of the exact confirmation varies, but you know, generally generating tens at least, maybe hundreds of gigabytes, maybe in some cases terabytes of data per day. And all of that data, so most institutions now will have fairly large data stores. All of this data will be written in native file formats. We can have a long discussion about what the nature of these file formats. John actually correctly identified one of the challenges is that these formats are defined and chosen by the vendors who make the instruments to be fast, right? They have to write very, very quickly now. And so the formats are write optimized, which is a completely legitimate thing to do if you're the guy who's building the stuff on the left, right? The problem is that almost all of our use cases are read, and that's turns out those are different, right? Okay, so we started circa about 2002 writing this library bioformats. It's cast in Java, and building readers, reverse engineering the file formats that we were seeing. And this was just sort of a labor of love of just receiving file formats from the community, people saying, hey, listen, I'm not sure that my current file format works with bioformats. Can you please test it? If it worked, we would tell them how to use it. If not, we would put it in the queue and just been sort of going through file formats. And so, and reverse engineering them, putting the readers into bioformats and releasing them online. Currently, there's a closing in on, I think, 160 different file formats and many different variants of these file formats of each of those. And so I don't actually know what we've not bothered to count what the actual number is, different versions of different file formats. But what's been great over the last few years is to see the commercial vendors actually getting on board and making contributions to this project, size Leica, and others. I would say that bioformats is more heavily adopted in the conventional biological imaging community than, for example, in digital pathology. One thing you should know, which is a slightly interesting, and happy to talk about this more, the team at the NCI Cancer Image Data Repository, so Andrei Fedorov, David Clooney, have commissioned bioformats, support for bioformats for there are several variants of DICOM WSI. So bioformats actually now reads and writes DICOM WSI in various formats. Okay. Happy to talk about that more. So think of it as a standard interface to everything on the left. So it's your one port of call to accessing wide variety of different file formats in many different domains. If you want, I can talk more about the strategy there and how that goes forward. Maybe you'll see as some things we're doing with formats at the end of the talk, we can come back to that. So bioformats is heavily used. It's in lots of open source software. It's in several commercial products, Zeiss's Zen software, OMERO, Columbus, and several others. So lots of people use bioformats. We put OMERO on top of bioformats, so that single logo is PostgreSQL database, a whole lot of permissions systems, and a whole series of API technology. And so the key there is that this is full-blown enterprise databasing for this type of imaging data. Yeah, Sean. So I think that's John's taking care of that, and what all John has to do now is support like another whatever it is, 140 odd file formats, and you're done. It's an interesting question, and you'll see where we're going in the end. Let's come back to that. Okay. So the short answer is not on your life, because those of you who have sat on grant panels, imagine me sending in a grant saying, I want to use competitive funding to fund a file format translator to deal with the panoply of file formats that are caused by chaos on the industrial side. That's it's a tough argument to win. We have gotten
very useful to like most of our work is all with like,
I know. Yeah, we'll get there. Yeah, let me come back to that because it's important. It's really important. But OMERO is think of OMERO as another set of APIs. I should say that when I talk to biologists, I will say, look, OMERO also has a lot of databasing capabilities in it. And that's important because one of the most important things databases do is control access. And so the illustration I make is, okay, when you get your airline tickets at your airline, you get yours and not mine, and I get mine and not yours. And the reason for that is there's a database. You can use the same illustration with banks, et cetera. And so for us, permissions, ownership, et cetera, is a key part. So row level permissions is a key part of any type of this type system. Okay, but OMERO is, again, APIs. So visualization tools, and in the early days, I know you guys don't think this was a big thing, but the idea that you could use a web browser to see multidimensional data was novel, and that detached accessing the data from where the data was stored. Okay, and so having support for multiple, wide number of viewers, our own and many others, but also importantly for this audience, analytics and processing. So APIs for C++, Python, et cetera, are all built into the system. I'm not going to tell you anything about the technology that's sitting under the APIs, but happy to get into the weeds if anybody cares. So that was the vision that we laid out when we started this project circa 2005. Sort of flagship papers published in 2012, and there's a whole series of publications around that, if anybody's interested. Okay, so sorry, so here you're going to see some screenshots of OMERO. This is from my laptop. So screenshots from my laptop connected through hotel Wi-Fi to various data here. What you're seeing here are the sort of standard 3-column layout, file organization on the left, thumbnails in the middle, and metadata on the right. Now, most of you have already flagged that this is DICOM data. It's a public DICOM data set. And the key point I want to communicate to this audience is we're pulling in the DICOM tags on the right-hand side. So this has to do with how OMERO is handling metadata. This is bioformats and OMERO working together to handle metadata. But bioformats in action, so the next slide, completely different modality, completely different metadata structure. This is high content screening data. So this is multi-well fluorescence imaging. And you see on the right-hand slide, that's actually cell profiler analytic metadata. So basically features for feature vectors coming out. So again, basically these interfaces working together to make all of this work. And I think the last example is TCGA data set. And some of you will notice the TCGA metadata on the right-hand side. And so it's basically exercising the capabilities of what these interfaces allow you to do. Okay? The only other thing I should have said that's built into the underlying data model is every image is a 5-dimensional structure. So space, time, and channel. So TCGA data is a 5-dimensional image with extent in time and channel of 1. Okay. All right. Yeah. The other thing that is also built into here is handling of pyramids, et cetera, which I won't really focus on, but is all built in inside. Okay. You can see. Yeah.
Does it support hyperspectral data then over time?
Yeah, so it's a really interesting question. So we have examples of supporting up to 8 different dimensions in the core data model, and that's effectively using modulo transformations into the unused dimensions. Not explicitly is the answer to your question, because every time we went down every time we went down multiple different paths, things there are too many sort of peculiarities to make something that's generic. However, each of the dimensions can be extended infinitely. There's nothing in the system that limits the extent of, for example, the channels. So hyperspectral data, geez, naturally goes into the multi-channel dimension. Okay, so that works. Okay. All right. Okay. So that's examples of OMERO. One slide on analysis, happy to talk about this more. But these are all examples of analytics, both open source and commercial, that connect to OMERO in various ways. this is, so we do not explicitly, we try not to get involved in building analytic solutions. That's lots of other people do that, but we're providing that foundation. And so that's, so these are all examples. And so I think there's on the website, there's documentation about how to make all of this work. These examples are all documented in one way or another. So, so who here hasn't heard of the acronym FAIR? Everyone have heard of FAIR? Okay. Oh, okay. So FAIR, so there was a paper published in about 2016, this idea that data should have these 4 different qualities, and that this was essential for data access. FAIR and the ideas around FAIR could be applied not only to public data, to private data. So, for example, data held in a private repository would need all of these qualities as well. Okay? So the data should be findable, accessible, interoperable, reusable. So FAIR. And importantly, there was definitions of what these words actually meant. But we think we're actually able to provide a lot of, so we think OMERO provides the infrastructure to make a lot of this possible. So it's been a sort of a guiding principle. Okay. So as I said, OMERO development starts circa 2005, first releases 2007, 2009, start having some success, and we've been moving on since then. In 2009, based on OMERO technology, Glencoe Software was actually contracted to build an image data repository for a journal that's quite prominent in cell biology called Journal of Cell Biology. And so Glencoe used OMERO and built one of the first, if not the first, BioImage data repository for one journal. And we got pretty large and actually was quite successful, and I'm happy to talk about that. But by having achieved all that, by sort of 2013, 2014, we started asking, okay, could we build a generic bioimage data repository for the world? And that led to what I'm going to tell you about now, the what we call the image data repository. And this is basically what we wanted to do. And we really wanted these data sets to be reference community resources, okay? And I'm going to define reference here in a second. Once you get into this business, you suddenly realize, and actually you guys know this very well, there are certain resources like TCGA, which are high because of their structure, their annotation and availability. All of these things come together, become extremely available. So, excuse me, become extremely valuable. But that's a lot different than throwing every H&E WS slide onto the internet somewhere and just letting people just fly at it, right? And so it's that well-constructed, well-annotated resource that we were really focused on. So that's what we've done. We've built this resource called the Image Data Resource, or IDR. We received BBSRC funding in 2015 to do this. Our first releases were in 2016, and all of this is what I'm going to tell you about now. So, so the IDR is running at EBI. It's running on their Embassy Cloud. It is a, guess what? Probably guess this. It's based on OMERO and bioformats, so it's probably looking familiar now. But our idea was to have as broad a range of data in the biological imaging space as we could. So that's what you see on the left-hand slide. On the right-hand side, have very rich annotations and curation. So that's what you see on the right-hand side, and use OMERO and bioformats to be able to do that. Obviously, we need a web-based access, so browsing, etc. We also wanted to enable cloud-based analysis because it wasn't clear. We thought that data download was not going to be how people were going to be able to use this data. That's turned out not to be true. Everybody wants to download terabyte scale data sets. And so in fact, there's a Globus and Aspera sitting behind all of this for those of you who want to download this data. I'll talk more about the cloud analysis in a second. So this was our goal to build this whole thing. And so what you're seeing here, the sort of roughly latest sort of vital statistics, about 360 odd terabytes of data, millions of image files, upwards of a little over 100 million image planes. Critically, because we have a lot of those annotations from 19 different organisms, 89,000 genes, okay, obviously across many different organisms. I'm going to mention in a second, we have original data from the Human Protein Atlas. So we have images from over 12,000 antibodies, and we have a large number of chemical screens as well. So lots of different types of studies are in IDR. Heavily used. So this is, I think, usage stats from, I think last year. Yeah, from last calendar year. About 34 million hits, about 78,000 IP addresses all over the world. So that's great. Okay, probably more importantly is to understand how we're working. So the first thing I want to highlight here is the number of studies we published, a little over 120, and I'm going to come back to that number in a second, with data associated with publications, and that's one of our sort of gate posts, right? So data has to be, except in a couple of exceptions, data has to be linked to a publication, okay? Because we feel strongly a peer-reviewed publication is the way we communicate in science. Separate discussion whether we like that or not, but in any case, peer-reviewed publication is the reference for a scientific report, and we're providing the data sitting underneath that publication. Okay.
That's a good question here, sorry.
Sure.
So in terms of, say, digital pathology images, how many sort of publications / sets of data have you actually got for?
Several. I'm going to give you a couple examples in a second. Anant Madabushi's cardiac biopsy data set is just one, and the whole HPA tissue data set is another.
Brilliant.
So, yes, I am.
So is it community led? So if I want to put my data there, for example, I need to pay, or how does it work?
So, okay, so to talk about this forever, but no, it's a really important question. So this is all grant funded, and the lessons. So I should say, we're following in the footsteps of great people who have set up GenBank, the genetic databases, then the genome databases, protein data bank, PDB, et cetera. And so we're very, so one of the lessons was, it would be great if you could charge people, but you can't. Okay, and so you have to find, so sustainability comes through grant funding, et cetera. And that's the first thing. And the second thing, which I feel quite strongly about, you have to have a fully open database, so no restrictions on, for example, commercial use, et cetera. I have lots of discussions with submitters about that. But the community, the user community is indeed the global scientific community, okay? But yeah, so our challenge is to keep this grant funded as well. And so the idea then is that, yeah, sure.
Usually apply for grant, OMERO, when then it makes it really hard for you guys to expand the data set, isn't it? How long does it usually take if I apply to ask the data that ask you to ask the data set? How long does it usually take?
How long?
Yeah.
The promise is in perpetuity. Yeah. So you're asking very important questions. We run the system at the European Bioinformatics Institute. There's a small number of institutions worldwide. We were talking about this at lunch, who are constituted, whose mission is to hold data for the community. One of them is EBI, another of them is in the United States, NCBI. In Japan, it's RIKEN. You could sort of count them on one hand. And actually, so Glencoe was part of PathLake. I think one of, I'll just speak out of turn here. Happy to be told I'm incorrect, but we're starting to think about these very large data projects, all of which are exciting and ambitious, et cetera. But, and I have to be careful here, but I can just tell you my university is not constituted to be a data resource. It's constituted to educate undergraduates, do research, et cetera, but it does not have the model in many, many different ways to actually serve data. Full stop. Maybe Warwick's different. I hope so. But so in that sense, actually, I'll just say, running OME at Dundee has been an interesting journey. Initially, they thought I was crazy, and definitely was crazy. But I think all of us are running up against the boundaries, the traditional boundaries of what our institutions are, how they're formed and how they understand what they're supposed to do. And so I do think, in a very serious way, we have to think about ways for those of us who are interested in large data projects. We have to contend with this issue. Fortunately, with EBI, they've been willing to host this data. And so here, maybe to your point, so I've worked hard, and please understand, this is not, I don't have 100% success rate, but I've worked hard to continue to fund IDR. But IDR data is all sitting at EBI, an institution who is constituted to serve data sets. I'll mention that in a second. And using open formats, et cetera. So if the worst possible outcome happens, IDR has to close down, that data is still there, it can still be used. Okay. So in some sense, our obligation to the submitters can be honored. Okay. So the idea here is that, in a standard peer-reviewed paper, there's a data availability section. Somewhere in there, there's an accession number, and that accession number is connected to our resource. Okay. Published lots of data, lots of journals, and all that stuff. Okay, so a couple quick examples. This is one of our flagship data sets. This is from Kate McDowell when she was in Philip Keller's lab at Geneva. This is the definitive fate map of the mouse embryo. This is a beautiful light sheet imaging, and all of this data is in IDR. This is the data set I just mentioned. This is from Anant Padabushi. This is their cardiac biopsy study. And again, it looks OMERO-ish, and yeah, that's the UI. And so, yep, hands up. It's not the most beautiful, but it does work. The thing I want to highlight here is the metadata that we've attached to this. So this comes in, this is metadata that we, as you can imagine, receive in spreadsheets that we then do all the curation with, okay? But so this was obviously, for those of you who know the study, this is a machine learning study. And so we have all of this metadata, and it's in it. Hopefully, this allows people to validate the study. Okay, human protein atlas. This is the tissue atlas. This amazing resource for those who have seen it, but all of the tissue data are available as JPEGs. The original data was sitting on tape archives and was not accessible because they didn't have any place to put it. Right, they're a scientific research institution, not a data repository, and so all of that data is now. So 12,000 of the 16,000 antibodies are now in IDR. This is all tissue pathology. This is all IHC staining, I should say. Okay. So one slide on ontologies. I won't really belabor this point, but we put a huge amount of effort into using controlled vocabularies around the various annotations. Happy to go into that in more detail should you care, but something we spent a lot of time on. Okay, a couple examples that I think are actually interesting and get to a few of the questions that we were discussing at this interface between tissue imaging and gene expression. So this is an in situ sequencing data set. There are both cells and tissues in this data set. So this is a case where you're using probes to identify the location of different genomic regions. So we have all the images you can imagine. I'm not going to go into the metadata here, but there's lots of annotations. But we also have, so the images, we have the region, the ROIs where each of the in situ spots are identified. And via the API, we have all of the annotations. So for every ROI, I think I highlight here. For every ROI on the left-hand side, we have the location, we have the UMI, we have the sequence, etc. So we can connect all of these data, and all of this is available via the API. So we published this data, and then we're very proud of ourselves, and then moved on. Jean Ma at Carnegie Mellon said, "Ah, that's pretty cool. I can actually make a 3D nucleome browser based on all of this data." And so they've actually published all of this. There's a really lovely paper in Nature Methods where they have an interactive tool that is pulling down all of this data. And so for us, this was validation that the API that we use can work. We've gone on. I doubt this is something across your guys' radar, but the US, there's a large project called the 4D nucleome, and they're coming up with metadata specifications. We've adopted all of that for spots, basically, fish spots. So, okay, so some examples, and now I want to go back to the fact that we only have 120. So I told you we started around 2016. We only have 120 sets, and that's because we publish reference images. These are these studies that are really broad. These are comprehensive studies across genes, across compounds. I haven't shown you any of the chemical studies, for example, SARS-CoV-2 inhibitors, et cetera, but all of that data is available. But what we're looking for are data sets across many different domains, as you sort of see there in the middle, but where there are, really, a broad sampling of gene perturbations, chemical compounds, cell lines, et cetera. So we're looking for data sets we think will be referenced and highly reusable for the future. So that's that's why we focus on a small amount of data. Now that raises the question about what about everything else? And so we've referred to that everything else as the bioimage ecosystem. There's a paper in Nature Methods a couple of years ago with our colleagues at EMBL that we published laying out this vision. You can think of IDR as an added value resource, or sometimes referred to as a knowledge base, basically an advanced integrated data resource. And for everything else, there needs to be data repositories, and so EBI is building the bioimage archive, and so that's the definitive home of most of the bioimaging data that's is associated with publications. All that's and that's going well, and we have a great relationship with Matthew Hartley, who runs that project. And so that's that's how we see this all developing. And so I think it's pretty important, if you're thinking about, if you want to build a digital pathology archive. So actually, I think this is where TCGA, runs into a little bit of trouble, because they try to do both. They try to be both the archive and the repository. And so what ends up happening is actually maintaining that level of annotation is hard at scale, and so then you stop. And so TCGA ends up freezing. And that's not a criticism, because it's a great project. But these are the realities that I think you have to really contend with when you get into this business. I would say that this isn't just our idea, that this is actually now an international consortium. So we work closely with the Systems Science of Biological Dynamics database at RECON, and so our colleague Shuichi Onami. And so we now see these national level, transnational level resources coming up all over the world, and so hopefully, this will continue. I will go back to, I mentioned learning from the previous efforts, in particular the Protein Data Bank. I'll just tell you this anecdote. Right before COVID, I was at a meeting in Los Angeles, and the speakers were invited to the meeting house for dinner. I was sitting on a sofa, and next to me, there was a woman next to me. She turns to me, and she's, we toasted. She said, "Who are you?" And I said, "Oh, my name is Jason Swedlow." She said, "Oh, you're that imaging guy, right?" And I said, "Yeah, you're Helen Berman." Helen Berman is basically, the parent, the mother, the father, whatever, all of them of the Protein Data Bank. For 50 years, she pushed through the PDB and made the PDB happen. And so we talked a little bit, and she said, "Yeah," she said, "it took us about 50 years to build the PDB. I hope it doesn't take you guys that long." And so the takeaway from there was, wow, okay, what a journey. It's also, she's an amazing scientist, incredible contribution. And think about what the PDB is to things like AlphaFold now, right? I mean, so that, those efforts come from great people. But I think, the key is that this, we're on a path, we're on a progression. And, that's certainly how genomics work. That's definitely how structural biology work. Hopefully it won't take us 50 years, but, it's going to take these multiple efforts at the national levels to make all of this. Okay, so Fayez said you wanted to submit data to IDR. That's how you do it. There's your email address. And, okay, so with all that done, and I'll try to get the last few slides really quickly. Question is where to go next. So one thing that IDR was doing, so some of you will have heard this expression, dog fooding. Is that something you've ever heard of? Yeah, some of you are smiling. Okay. This idea of eating your own dog food, using your own software. Okay, so using bioformats and OMERO and IDR, we learned a lot about this. So what they could and couldn't do, and where they really fell apart. And one key problem about all of this was how to work in these very large resources, okay, and how to think at scale. And so that really raised this issue. This is a word that will be familiar to many of you, but the question is, how do we work at the scales that are necessary to be building these large global repositories? And many of you know that when you start working in these architectures, many things change quite dramatically, and a lot of the assumptions, the basic assumptions, work quite well in our laptops don't really hold. And certainly one of them has to do with storage and how data is actually laid down on this. And just the storage representation of data is completely different in cloud-based resources. And so we started talking about this circa 2017 as we started seeing IDR grow. And around 2018, 2019, decided that we had to do something about this, that our strategy for handling data was not going to be sustainable. And in particular, having taking proprietary file formats in file structures that were designed for POSIX file systems and doing bioformats mediated real-time translation was just not going to work. And we were starting to see IDR choke on that strategy. And so that's when we started dialing back on our efforts actually to continue real-time translation with bioformats, because we don't think that real-time translation is sustainable. Okay? We need something else. Okay? So that's something else. That's what we call OME-NGFF. And so it was probably a mistake to call it the next generation file format, because a few years later, it still is the next generation file format. But the idea, we picked up technology that Alistair Miles at Oxford had developed, this idea of a chunked data representation that was better, more optimized for example, cloud-based stores, object, S3, whatever you call them. And also, this is critical, could also hold metadata. Okay, so we could align the metadata right with the binary data. So now, this is the kind of thing that gets very religious quickly, but we start to think about serverless implementations, et cetera, which may or may not be relevant. We start to think about data streaming, et cetera. Okay, and so we could. So as I mentioned, we had seen in IDR a lot of people saying, "Well, I want to download the data." Well, when we're holding terabyte scale data sets, actually, that's probably not the best idea. We need some sort of streaming application. And so Napari now supports ZAR directly. And so data can be loaded on S3 in a ZAR representation and accessed directly, and all of that works. That's not to say that that is completely without limitations. Like all great technologies, there are things it's good for, things it's not good for. But it does allow us to think about serverless implementations, streaming, et cetera. And so we've done all of that. What we haven't yet done, but I think we really need to think about doing, is federated data resources. And so at least the vision is there for doing that. I can tell you the reviewers have all hated that vision, but we'll get there. It'll happen. It has to happen. Going back to the discussion, can't, all data generated by all members of the community, it can't live in a single location anymore. That was fine for,
Streaming is always fine. When you try to download, it's much slower because there are chunks of.
Yeah.
Of course, how do you suggest that?
So the short, you sort of know the answer already. There's no single answer to these questions, but there's obviously options. On the data storage side, there's ideas about sharding. There's ideas around zipping data up in various ways, and all of those are options. And there's now support. There's now published support or available support for sharded ZAR. So that's one idea. But the other idea is, well, one is to download the data, and there are people who are doing that. It would have to be in a zipped representation or something like that. And finally, then there's portable models, and there's moving the models to the data. And none of those are all different ideas, and there's various people doing those different things. You guys are much more knowledgeable about all of that than I am. And I think, like all the answers to all of such problems, there's not going to be a single answer. It'll depend on the details, but it's going to be one of those. So we were talking a little bit about this over lunch, and I'm aware of the time's passing, but I personally think I think we have a problem to solve with the commercial clouds. Great corporations, great companies, building solutions for Word documents, which is absolutely fine. There's nothing wrong with that. But those solutions, in particular their pricing, are not constituted for research data. They're not constituted for academic research data. But I can tell you from experience with Glencoe software, they're not constituted for industrial data either. I mean, the number of cloud deployments of digital pathology data we've built a proof of concept that are working is numerous, and almost every time, not every time, but almost every time, company that says, well, that's nice, but look at the cost. Basically, access requests and egress are too high. Forget it, we'll bring everything on premise. And so how is it that these organizations don't see that there's so this is there's a commercial success to be had there. It's a deep, serious problem if you think about the importance of the problems we're working on for diagnosing any of the diseases we're talking about. So I think something has to be done there. In the meantime, I was saying at lunch, just one thing. I personally think, there's some amazing resources, national labs here and in the US. I personally think we need to avail ourselves of those until the commercial guys can't get that. Sorry if I was.
How they transfer ID or dashboard?
Well, so we have a Jupyter system all hooked up to IDR, and we have a whole bunch of Google Colab notebooks connected into IDR. So yes, we've done that. The problem is just it's a scaling problem, and no matter what you do, everybody wants more. So everybody wants, well, now I want 100 GPUs and so on and so on. And I think I personally think, I mean, you're talking about global resources. I think national level computing resources exist. They really do exist. So why aren't we making better use of them? I'm not sure that that's the long-term solution, but I can tell you in my LEAP side, I mentioned to some of you, we built data repositories at the San Diego Supercomputer Center, or a supercomputer center, right? They really don't even bat an eyelid at 50 petabytes. Right? It's just, if you say 50, if you say a petabyte and 500, and a large HPC solution, they're like, yeah, sure. I mean, we can have that up tomorrow. What's the problem, right? So I think that's what we're going to do, right? Okay. I rattled on long enough. All of the software for NGFF is open source. It's great to see John's work with this, knocking us off the block. So that's great. I will take that home. I will say the one thing. So this paper is now published. So we came out with our first paper just with the ideas and some benchmarking, et cetera. This is sort of an update a year later about adoption. And the coolest thing about this is that I think it's Journal of Statistical Chemistry and something. Anyway, just look up OME-TIFF, you'll find it. The organizations represented in the author list are really interesting. So global, all sorts of different types of organizations, small research institutions, the largest research institutions, corporations, et cetera. So this is really exciting to see this level of adoption, and yeah, looking forward to taking it forward. Okay, so told you about some file formats in OMERO, IDR, NGFF. I think, we think going all the way back to the underlying data representation is important for setting up this next generation. That's what we're doing. I think what's been really fun is working now at a global community with open source teams, imaging networks, imaging infrastructures, et cetera. So I think this community, starting out with a few of us in Dundee, and the community's been growing in lots of places, including, for example, here. I think that's been, I have to say, one of the most exciting aspects of all this. Final slide, an amazingly talented and dedicated team doing all of the work. And so thanks to them. And in particular, thanks to the funders, in particular, the Welcome, the BBSRC, who have funded a lot of the OME and IDR work. And with that,
Fantastic. Thank you for a really interesting talk. So with that, I suppose we'll does anyone have any questions at all? Yeah.
Thank you very much for waiting.
I missed the
full-size scanner manufacturer use NGFF file format. So, I mean, when you generate full-size, you generate them in NGFF.
Right. So I think you shouldn't ask me, you should ask John, because probably no one knows more about writing these formats than John at this point.
For us, that would be great. I don't think it's going to actually like it because they want to optimize good getting things on bit scale as quick as possible.
Yeah.
Because they want to have smaller buffers and cheaper. Because I mean, if you have memory buffers in the hardware, that's expensive. They want things as small as possible to get it down the disk really quickly. So I think there's competing interests on what the big format plus. That's what's good.
Yeah. So for the moment, and I don't know how long the moment lasts, but it's going to last a little while. I think it's likely. So we've had very open and very direct conversations with many of the different microscope or imaging system manufacturers. This is a read optimized format. This format is for your use cases and it's not for theirs. But look at what we did. We scanned the horizon. We talked to a whole bunch of people. We tested a whole bunch of technologies. There's many others we could talk about. And then we engaged with our community and we tried to and we came together to do something. Well, you guys could do the same thing too. And oh, by the way, sorry, you guys, the vendors could do the same thing too. And oh, by the way, you have the use cases. You know your requirements much better than we do. And as John says, I mean, you know how much memory you're willing to put on board, et cetera, and all of that buffers. So you should do this. If you think we're just going to do it for you, I can't fund that. But more importantly, we're the wrong people to do that. Right? And I think, ever since I've been in imaging, the industrial guys have just been, they've competed, to the death. but I think, you know, DICOM's an example of vendors coming together and say, okay, we'll do something pre-competitive for our you know mutual benefit. And I imagine that for example, the digital pathology vendors or any of the other teams could come together and do exactly the same thing. For what it's worth, this problem is universal across detectors now, and it's not just, you know, the space.
Brilliant. So do we have any more questions at all from the group?
Just have a general question, and I apologize in advance for the question.
No.
It's not nothing to do with OMERO or what we're doing, but starting off with a chemical background in chemistry and journeying to something very computer sciencey, file formats. Understanding that and building a company of that and building enabling technologies. So we have our PhD students there. What lesson would you give across the range to have a range like yours?
Well, I think it's, well, first of all, I don't think anybody should necessarily aspire to do whatever. You should follow what you think is right, but maybe that's, I think, you know, each of the steps I've told you about, you're confronted with a problem and it's just like, I have no idea at all how to think about that problem at all. And no one else can help me out either. Okay. Well, that'll probably be a space of, you know, something that, you know, and I should say, do I think it's important? Engaging that is a tricky one. But I think, you know, problems where there's just a lot of territory to work in, are often good spaces. I guess maybe the only other thing I would say, and this is really important to understand OME. OME is a group of incredibly talented and committed, but slightly crazy and a little bit odd individuals, include me in that. And it's one of these things where together we're much better than our parts. And so, and we've worked together now for over 20 years. And so I guess the other thing to say is see if you can find a partner. And you'll need, because, you know, good partners and good collaborators, people who you want to listen to and who you trust, you know, will let you do more, will get you beyond your own limitations. I don't know if that's helpful. I have no idea if that's helpful. Good.
Fantastic. Well, thank you very much.
Yeah. Sorry for going over. And thank you again, John. Thank you. Thank you, Sean.
I think, well, thank you for everyone.
Say hi to Nazir.
Yeah, it's great to talk, Jason. Thank you, and sorry again that you didn't join.
No problem. Mark is no different, I'm not sure.